Hi everyone, I'll be presenting on the topic what makes the beta hairpin from B1 domain of protein G extra stable. As the title suggests, why GB1 hairpin? It governs the formation of protein G, which is an immunoglobulin protein, and also it's a motif that is stable even at microsecond time scale. So given that it is highly stable, it's important to study the stability of GB1 hairpin. Yeah. Now the question arises, what defines the GB1 hairpin? So we carried out simulation and we found that there are 7 backbone hydrogen bonds present in GB1 that defines the unique folded state. We also found that there are 7 side chain hydrogen bonds that provides additional stability. You can see the probability distribution curve of backbone and side chain hydrogen bonds. So during the entire course of the simulation, we found that our protein was in a folded state. So we were in a confusion, like whether it's trapped in the global minima. So in order to ensure complete sampling, we performed a replica exchange molecular dynamics simulation with a temperature range of 300 and 477 Kelvin. And the trajectory at 300 Kelvin was extracted and the hydrogen bond distribution calculation was performed. As you can see from the graph here, that the distribution, hydrogen bond distributions obtained here was consistent with the one obtained for classical MT. Now, as I already said, there are 7 backbone hydrogen bonds and 7 side chain hydrogen bond. But is that all? Are there any other factor that contribute to the stability of GB1 hairpin? So in order to find an answer for that, we performed simulations with and without mutations in the presence and absence of urea, which is a protein secondary structure denaturant. Yeah. So you can see this is a probability distribution curve for the RMSD of proteins. So as I already said, GB1 hairpin is highly stable in aqueous solvent. So what we did is that we put GB1 hairpin in urea. As you can see from the black curve, it was highly stable, which means urea was not able to act as a denaturant for GB1 hairpin. Yeah. So why it is so stable? So in order to find an answer for that, we mutated the hydrophilic groups. As you can see from the graph here, when the hydrophilic groups are mutated, the number of urea molecules around the protein backbone increases, which causes the denaturation. And with around 50 nanosecond, you can see from the yellow curve that the protein got denatured, which means it was highly unstable. Now, the same hydrophilic mutated protein, it was added in water, not in urea. And as you can see from the red curve here, that when the hydrophilic mutated GB1 hairpin was put in water, it was highly stable, which is quite shocking because even though the hydrophilic groups were mutated, it doesn't affect the stability, which means there are some other factors that contribute to the stability of the GB1 hairpin. So what should be? So in order to find an answer for that, we mutated the hydrophobic groups. So as you can see from the blue curve that we put the GB1 hairpin, hydrophobic mutated GB1 hairpin in just plain aqueous solvent, not in urea. And from the blue curve, it's highly evident that at around 22 nanoseconds, the protein got denatured, which is quite a finding. So I conclude by saying that the backbone hydrogen bonds are responsible for the unique fold of GB1 hairpin, and there are 7 side chain hydrogen bonds. They provide additional stability to the hairpin, and the key factor holding the beta hairpin is the hydrophobic groups, which also gives extra stability to the hairpin. Without the hydrophobic groups, the protein crumples altogether. Thank you.